Delighted to be opening another Global Immunotalk. I'm very pleased to be connected with our co-organizer, Carla Rodlin, and our amazing speaker and dear friend, Dr. Gabriel Rabinovich from Buenos Aires, Argentina. So before Carla introduce Gabi, I would like to briefly remind you, as always, about the goals of the Global Immunotalks, which are to benefit and inspire immunologists across the world in an egalitarian manner and without traveling, making these talks accommodating, inclusive, and environmentally friendly. And remember, the talks are free, are recorded in YouTube, and the discussions are virtual, so the Q&A is via Twitter. As always, we are very thankful to all the speakers from different nationalities, different parts of the world who have contributed to this initiative. And we would like to remind you that next week, our global immuno speaker is Laura McKay from Australia. So Carla, you can take it away then to introduce Gabi. Thank you.
Thank you so much, Elena, and it is indeed my real pleasure to introduce today our global immuno speaker, Dr. Gabriel Rabinovich. So Gabriel is from Córdoba, Argentina, and that's the very same province where our co-organizer, Elena, is. So indeed, they have many things in common. Gabriel graduated with a degree in biochemistry at the University of Córdoba. And he also obtained his PhD at the same university. The PhD was in immunology and was under the direction of Dr. Clelia Riera, a pioneer immunologist in Argentina who trained a series of current immunologists. He then moved, but not too far, to the capital city for a postdoctoral position at the University of Buenos Aires. Gabriel quickly rose through the ranks, and he is currently a senior investigator at the National Research Council, as well as a professor of immunology at the University of Buenos Aires. Gabriel heads the Laboratory of Immunopathology at the Institute of Biology and Experimental Medicine, known as the IBIME, which is in the same building where I did my own PhD, so it's a place that is very dear to me as well. Now, Gabriel's work has centered on the role of glycoproteins, more specifically galectins, and their role in the regulation of the immune response. His laboratory has shown that galectins can function as what he calls glyco-checkpoints, and in doing so, they can limit inflammation, suppress autoimmunity, and promote fetal-maternal tolerance. Now, as you can imagine, the flip side of these glyco-checkpoints is that they might be able to allow cancer cell growth and invasion of microbes. Based on his outstanding work, Gabriel has received numerous awards and recognitions. I will only introduce you to some of the highlights, which have been, for instance, being elected to the Argentinian National Academy of Science, as well as the National Academy of Science in the United States. He also received the Investigator of the Argentinian Nation Award in 2017. So I think today we're going to learn more about this fascinating biology through his Global Immunotalk entitled The Sweet Escape, Glyco-checkpoints as therapeutic targets in cancer, autoimmunity, and infection. Gabriel, it's a real pleasure to have you. Thank you so much for accepting to present at this forum.
Thank you very much. Thank you very much. Carla, can you hear me well?
Yes, we can hear you very well.
Great. So thank you very much, Elena. Thank you very much, Carla, for this invitation and this kind introduction. It's a real pleasure, but a real pleasure to stay here to speak in this prestigious meeting, in the prestigious series of seminars. I feel really honored and privileged to stay here. So thank you very much for this invitation.
Gabi, thank you so much for doing this. Now, as you know, we like to get to know our speakers a little bit better. So we have a question for you. As you well know, Bernardo Say, who was a Nobel laureate from Argentina and considered the father of physiology in Argentina, is very well known for saying that science does not have a homeland, but scientists do. And you seem to have followed in his footsteps as you trained and established your independent laboratory in your homeland, Argentina. Now, we all know sometimes the resources can be a little bit more limited, but you have had a stellar scientific career, all made in Argentina. So can you share with us some of the highlights of running your lab in your home country?
Okay, thank you very much, Carla, for this question. In fact, probably it's the question of my life. Yes, when I finished my PhD, like all the young scientists, I thought to travel abroad for a postdoc, and in fact, I applied for a Pew Fellowship to travel to the NIH, to the US. Finally, for personal reasons, I couldn't make it. And at the very beginning, I was a little bit disappointed and I wanted to travel, but then I thought with idealism that I could fulfill my dreams to try to close the circle of basic research, biomedical research, and clinical applications of our findings in Argentina, in my country. So I felt really very proud of doing so, and there are some advantages and disadvantages. So Argentina has a tradition of science, like you mentioned, with Bernardo Cusay, with Federico Leluar. Also, Cesar Milstein was Argentina, who was a great immunologist and a Nobel Prize. So we are very proud of the tradition of science in Argentina. And also there is a lot of young talent scientists that work very hard. So these are the advantages. And the disadvantages are the disadvantages of the region, in general, Latin America. So there are a lot of political, social, and economical, mainly economical turbulence. And, you know, science mirrors all these political and economical crises. And we always face low salaries and low budgets, low grants, small number of grants. And also, you know, the problem also is the bureaucracy to try to get reagents, to import reagents, and to import. Sometimes we need to wait 2 or 3 months until we get an antibody to finish an experiment. This is a little bit frustrating. But at the end of the day, to every 2 or 3 years, I think, did I do the right thing or the wrong thing? But then I recover the idealism to go on and to do things in Argentina. And I like to stay with my group, working very hard to do the best science we can.
Thank you. Thank you so much for sharing that perspective. And thank you for your hard work in Argentina and for training the next generation of Argentinian scientists.
Thank you.
So now I think we're ready to listen to your talk. So you can start sharing. Okay, and perfect. It all looks good, Gabriel.
Great. So today, as Carla mentioned, I will speak today about glyco-checkpoints, therapeutic targets in cancer, autoimmunity, and infection, what we call the sweet escape. I will summarize some previous findings and novel findings that characterize the role of galectins as glyco-checkpoints. So during the past decade, we have witnessed a revolution in cancer immunotherapy, mainly associated with the discovery of immune checkpoint pathways, particularly those triggered by CTLA-4 and PD-1, and the implementation of checkpoint blockade therapies, which improved overall survival and progression-free survival in a great number of patients by unleashing anti-tumor T-cell responses. However, several patients do not respond to this treatment, highlighting the need of additional immunotherapeutic approaches. But what are immune checkpoints? Immune checkpoints are inhibitory pathways, co-inhibitory pathways that limit the duration and amplitude of immune responses. They operate both during the induction phase and during the effector phases of an immune response and contribute to create immunosuppressive microenvironments by targeting different immune cells, including cytotoxic T cells, regulatory T cells, macrophages, dendritic cells, NK cells, myeloid-derived suppressor cells, and even fibroblasts, and endothelial cells. There are 4 hallmarks that characterize an immune checkpoint. They are often caused by tumors as a mechanism of T-cell resistance. They promote resolution of autoimmune and chronic inflammation. They contribute to immune tolerance at the fetal-maternal interface during pregnancy, and they control the resolution of microbial infections and allergic reactions. But I would like to introduce today the concept of glyco-checkpoints. Although originally conceived as glycobiomarkers that delineate the prognosis of different tumor types, this includes TN, sialic acid, recent evidence suggests that glycans and glycan-binding proteins can also serve as glyco-checkpoints that suppress anti-tumor T-cell responses. To illustrate this concept, sialylated antigens, including sialyl T antigen, sialyl Tn antigen expressed on tumor cells, can be recognized by Cglex, which are lectins expressed on innate immune cells. Also, galactosylated antigens, such as Tn antigens or GalNAc, can be recognized by the C-type lectin MGL, the macrophage galactose lectin, and fucosylated antigens can be recognized by innate immune cells, such as DC-SINE. And all these recognitions, all these interactions between glycan-binding protein, even Cglex, C-type lectins with glycosylated structures, can suppress anti-tumor immune responses. But we will focus today on galectins. Galectins are a family of soluble glycan-binding proteins that recognize preferentially the disaccharide N-acetyl-lactosamine. N-acetyl-lactosamine is galactose beta-1,4 N-acetylglucosamine, so this is a disaccharide present in complex and glycans and also in core glycans. They are highly conserved throughout the evolution from fungi to mammals, although there are some galectin-like folds even in viruses. In response to stress or inflammation, galectins are secreted to the extracellular compartment through an unconventional Golgi independent pathway, which has been a mystery for many, many years. In the last part of my talk, I will present you some very recent data that is impressed that we performed in collaboration with Vijay Rathiman that can provide some clues on the mechanisms of galectin secretion. Galectins are classified into 3 different subfamilies: the prototype galectins, the chimera type galectins, and the tandem repeat galectins. We will focus today on the prototype galectins, particularly galectin-1, which has 1 carbohydrate recognition domain that can dimerize. Extracellularly, galectins can form a kind of network of lattices that are multivalent interactions with glycosylated receptors, and in this way, by forming these galectin carbohydrate lattices, it can control receptor segregation, endocytosis, and signaling. So in early studies, we identified galectin-1 as a 14-kDa beta-galactoside binding protein expressed in inflammatory macrophages. It showed broad immune inhibitory activity, suppressing T-cell activation, pro-inflammatory cytokine production, and survival. What was very interesting was that galectin-1 expression was elevated in tumors, and the expression correlates with the acquisition of a metastatic phenotype of these tumors. Analysis of TCGA data reveals that galectin-1 expression is associated with poor survival in most human tumors analyzed, as you can see here. So, given the expression, the high expression of galectin-1 in the tumor microenvironment, and its broad immune regulatory and immunosuppressive activity, we propose that galectin-1 may serve as a glyco-checkpoint that promotes tumor immune escape, that allows tumors to evade anti-tumor immune responses. In fact, we found in the B16 melanoma model that galectin-1 contributes to the immunosuppressive activity of melanoma cells. When we blocked Galectin-1 expression, we found a reduction of tumor growth, and this effect was partially prevented when we depleted either CD4 or CD8 positive T cells, and was fully prevented when we eliminated both CD4 and CD8 positive T cells. We found that silencing Galectin-1 gene expression in tumors induces an interferon gamma dependent Th1 and cytotoxic T cell responses in vivo. We confirmed these findings in human classical Hodgkin lymphoma in collaboration with Margaret Schiff from the Dana-Farber Cancer Institute in Boston. In this particular model, we found that expression and secretion of Galectin-1 by Ries-Steinberg cells contributes to create a Th2-skewed immunosuppressive microenvironment in classical Hodgkin lymphoma. At the molecular level, Galectin-1 was found to be upregulated in Ries-Steinberg cells by an AP1-dependent enhancer controlled by Epstein-Barr virus, and more importantly, Galectin-1 serum levels can reflect clinical outcome in this tumor type. So based on data from our lab and also from other laboratories around the world, we now know that Galectin-1 activates immunoevasive programs in several tumor types, including glioblastoma, T-cell lymphoma, breast adenocarcinoma, pancreatic adenocarcinoma, ovarian carcinoma, Kaposi sarcoma, chronic lymphocytic leukemia, classical Hodgkin lymphoma, as I mentioned, lung adenocarcinoma, and neuroblastoma. So in most tumor types, Galectin-1 plays a role in tumor immune escape. But we wanted to understand the molecular mechanisms that control the immunoregulatory activities of Galectin-1. So for this, we first decided to dissect the glycosylation signature of different immune cell types, or in other words, like we tried to express in this review that we wrote for immunity, to decipher the glycocodes in immunity. So as I mentioned, Galectin-1 interacts with lactase factors, here highlighted in yellow and blue, galactose, beta-1,4, and acetylglucosamine. You can see here the structure that are present in complex glycans and core-glycans. So the more lactosamine structures you have, the higher affinity of Galectin-1 to these glycans. There are two glycosyl transferases that are critical for Galectin-1 binding. One is the MGAT5 glycosyl transferase, and the second one is the C2GNT1. MGAT5 contributes to beta-1,6 and glycan branching and elongation of lactase structures, and C2GNT1 contributes to core-2O glycan extension and also elongation of lactase structures. However, alpha-2,6 sialic acid generated by the glycosyl transferase ST6-Gal1 blocks Galectin-1 binding. So what we need to remember, MGAT5 and C2GNT1 are important for generation of galectin-specific ligands, and ST6-Gal1 is important for disabling Galectin-1 interaction to different immune cell types. So when we explore the T cell compartments, we found that during differentiation, Th1, Th17, and CD8 positive T cell express all the repertoire of glycans that are important for Galectin-1 binding and induction of cell death. This includes higher expression of beta-1,6 and glycan branching, higher core-2O glycan extension, and lower levels of alpha-2,6 sialic acid, so Galectin-1 can bind to these cells and induce apoptosis or dysfunction of these cells. In contrast, Th2 cells, and now we know that also regulatory T cells, are covered by alpha-2,6, by high amounts of alpha-2,6 sialic acid, which prevents Galectin-1 binding to these cells. So this effect was confirmed by single-cell RNA-seq analysis of T cells from melanomal biopsies, which revealed correlations between gene expression of glycosyl transferases and cytotoxic T cell markers. In fact, increased expression of cytotoxic markers such as granzymes and CD8 positive and the CD8 marker correlated with reduced expression of ST6-Gal1 an increased expression of C2GND1, allowing Galectin-1 binding. But we wish to explore the role of Galectin-1 within the regulatory T cell compartment. So in a model of triple-negative breast cancer, we found that although Galectin-1 does not kill regulatory T cells, it promotes expansion of FOXP3 positive regulatory T cells at the systemic level, promoting lung metastasis of these breast carcinomas. So because of the importance of dendritic cells in orchestrating immune responses, we also wanted to dissect the glycosylation signature of dendritic cells and to explore the role of Galectin-1 glycan interactions within the dendritic cell compartment. And what we found was that Galectin-1 binds particularly to O glycans on CD43, promoting tolerogenic dendritic cells. These tolerogenic dendritic cells produce very high amounts of IL-27. IL-27 bind to its receptor on the cells, promote differentiation of IL-10 producing regulatory type 1 cells. So we identify an immunoregulatory circuit that was mediated by Galectin-1, IL-27, and IL-10. So supporting these findings, the tolerogenic effects of Galectin-1 on dendritic cells were eliminated when we interrupted the IL-27 or the IL-10 pathway as measured by interferon gamma production, IL-17 production, and IL-10 production. And this effect in the tumor microenvironment was governed by the SATB1 transcription factor. So one of the most consistent findings in our work was that when we blocked Galectin-1 expression, there was not only an increased anti-tumor immune response, but also a reduction of blood vessels. Because of the functional crosstalk between immunosuppressive and pro-angiogenic programs in the tumor microenvironment, where many different pro-angiogenic mediators also trigger immunosuppression, and whether other immunoregulatory cytokines also promote angiogenesis, we wonder whether Galectin-1 glycan interactions may also control angiogenesis. In fact, we found that Galectin-1 indeed promotes angiogenesis, and this effect was prevented when we used a Galectin-1 mutant in the carbohydrate binding site, or when we knock down MGAT5 on endothelial cells, disabling the production, the generation of beta-1/6 and glycan branching. So when there is no Galectin-1 or when there are no complex in glycans. This effect was confirmed in vivo in a Kaposi sarcoma model. So we wish to identify a possible receptor for Galectin-1 on endothelial cells and a possible signaling pathway that could mediate the pro-angiogenic effect of Galectin-1, and we perform RTK signaling arrays. Surprisingly, we found that Galectin-1 binds to the same receptors as the canonical cytokine BGF. So we trigger phosphorylation of BGF receptor 2, phosphorylation of AKT, and phosphorylation of L1Q to promote angiogenesis. These effects were interrupted when we silence BGF receptor 2, of course, but only when we silence MGAT5, we could block MGAT5. Remember, it's the glycosyl transferase that generates glycan branching. We could abrogate the effects of Galectin-1 on endothelial cells, particularly for correlation of BGF receptor 2, AKT, and L1Q. So only when we blocked BGF receptor 2, but not when we block other members, other BGF receptors, such as BGF receptor 1, 3, integrins, or other glycosylated receptors on the surface of endothelial cells, we could block tube formation induced by Galectin-1. And the question was why? BGF receptor 2 is a glycosylated receptor that has 7 immunoglobulin-like domains and 18 N-glycosylation sites. So we did site-directed mutagenesis of this receptor, and we found, with these experiments, that Galectin-1 binds selectively to immunoglobulin 3, 4, and 7 domains. Also, we found that particularly BGF receptor 2 was highly covered by beta-1/6 and glycan branching and low levels of alpha-2/6 thiolic acid. In contrast, BGF receptor 1 and 3 were highly covered by alpha-2/6 thiolic acid, and this explains the selectivity of galectin-1 for BGF receptor 2. So, in summary, galectin-1, in response to hypoxia, galectin-1 is released from tumor cells through a HIF-independent and NF-kappa-B-dependent mechanism. Galectin-1 then binds to BGF receptor 2 to complexing glycans from BGF receptor 2 on endothelial cells and promote BGF light signaling, mimics BGF light signaling to promote angiogenesis. So based on these findings, we asked the question whether galectin-1 glycan interactions may preserve angiogenesis in anti-BGF resistant tumors. So when we block BGF, still galectin-1 can preserve angiogenesis through binding to the same receptor as BGF, particularly in those patients that are resistant to anti-BGF monoclonal antibodies. So we performed an in vivo experiment in which we inoculated tumor cells that are resistant or sensitive to anti-BGF treatment. We treated these mice with anti-BGF and analyzed galectin-1 expression at different days on tumor cells in the tumor microenvironment. We found, particularly at day 4 after treatment, and in other tumors also at day 7, and this is important because day 4 is the day of tumor hypoxia following treatment with anti-BGF in vivo, we found an upregulation of galectin-1 expression only in tumors that are resistant to anti-BGF, but not in tumors that are sensitive to anti-BGF. And this was specific for galectin-1, as we couldn't see any effect with galectin-3, galectin-8, or other members of the galectin family. So we did the same experiment, but analyzed the glycosylation signature of endothelial cells in vivo. So we inoculated tumors resistant or sensitive to anti-BGF. We treated these mice with anti-BGF monoclonal antibody, and then we purified endothelial cells that were associated to tumors. And what we found was that endothelial cells associated to tumors that were resistant to anti-BGF, but those that were sensitive to anti-BGF had lower levels of alpha-2/6 thiolic acid and higher amounts of beta-1/6 and glycan branching, which allows and favors galectin-1 binding. So after anti-BGF treatment, not only there is an increased galectin-1 expression in tumors, but also the doors are open for galectin-1. So endothelial cells express all the glycans that are important for galectin-1 binding and angiogenesis. So we performed the same experiments, but we knocked down galectin-1 in tumor cells to determine whether galectin-1 glycan interactions really are important in promoting resistance to anti-BGF treatment. And when we silenced galectin-1 expression, we successfully eliminated resistance to anti-BGF treatment. So those tumors that were originally resistant, they were now sensitive to anti-BGF treatment. And this effect was even more pronounced in one knockout mice. So we did the same experiment, but on the other side of the street. We eliminated selectively beta-1/6 and glycan branching on endothelial cells, so galectin-1 cannot interact. And again, those tumors that were resistant to anti-BGF treatment, they now became sensitive to anti-BGF treatment. There is no beta-1/6 and glycan branching, so galectin-1 glycan interactions cannot compensate. On the other hand, when we performed the other experiment, the opposite experiment, we inoculated tumors that are sensitive to anti-BGF in FT6-GAL1 knockout mice, in mice lacking alpha-2/6 thiolic acid on endothelial cells. And in these particular models, tumors that were sensitive, such as the B16 melanoma, that were sensitive originally to anti-VEGF, they became refractory. There are no alpha-2/6 sialic acid, so Galectin-1 can interact with endothelial cells. So in summary, in tumors that are sensitive to anti-VEGF treatment, anti-VEGF treatment induces high amounts of alpha-2/6 sialic acid on endothelial cells, so Galectin-1 cannot interact and cannot promote angiogenesis. In contrast, in tumors that are refractory to anti-VEGF, resistant to anti-VEGF, endothelial cells are covered by high amounts of beta-1/6 and glycan branching, low amounts of alpha-2/6 sialic acid, which allows Galectin-1 binding to immunoglobulin 3, 4, and 7 domain, promoting VEGF-like signaling, revascularization, and allowing escape from VEGF blockade. So we wanted to integrate the immunoregulatory activities of Galectin-1 and the pro-angiogenic activities of Galectin-1. And for this, to achieve this goal, we started a collaboration with Pilar Navarro, Hospital del Mar in Barcelona. Why we choose pancreatic cancer adenocarcinoma? Because it has the lowest survival rates, the poorest prognosis, and they are resistant to all kinds of immunotherapy. So in this particular tumor type, Galectin-1 expression is upregulated in human pancreatic cell itself. So we use a transgenic model that recapitulates all the features of human pancreatic adenocarcinoma, and we cross these mice into a Galectin-1 background. So we use this mouse transgenic model, and we also use a human cell-based system in which we co-culture human pancreatic cells with immune cells, but also with tumor cells. And we found, as a result of these two models, that inhibition of Galectin-1 contributed to a reduction of tumor metastasis, particularly to the liver, a reduction of angiogenesis, of migration of tumor cells, a recruitment of CD4 positive and CD8 positive anti-tumor T cells, and also a reduction of CD11B positive, GR1 positive myeloid-derived suppressor cells. So myeloid are highly enigmatic, as you know, it's a highly enigmatic, immature, and heterogeneous cell type in the tumor microenvironment, and also inflammatory and infectious, particularly a pro-angiogenic and immunosuppressive phenotypes that contribute to tumor progression. So we wanted to examine the role of Galectin-1 glycan interactions in myeloid-derived suppressor cells. We found that we dissected the glycosylation signature of monocytic and granulocytic myeloid-derived cells, and we found that Galectin-1 is a common denominator that hierarchically regulates immunosuppression and angiogenesis in the tumor microenvironment, and the pro-angiogenic and immunosuppressive activity of myeloid-derived suppressor cells that contribute to tumor progression. But one of the most important questions is, how can we target Galectin-1 expression in the tumor microenvironment? So we first studied by designing glycoamines, lactulosamine, which blocks Galectin-1 carbohydrate binding domain, but also other members of the Galectin family. And then we moved to small molecule inhibitors. But finally, we designed and generated different types of anti-Galectin-1 neutralizing monoclonal antibodies. What is the advantage then of blocking Galectin-1? It stimulates anti-tumor immune response, it unleashes anti-tumor T-cell responses, but also blocks tumor angiogenesis. So we recently generated and published neutralizing anti-Galectin-1 antibody, which was highly specific of Galectin-1. It blocks and neutralizing Galectin-1, but not other members of Galectin family. And this is particularly because it binds to an epitope which is outside the ligand binding group. It is highly specific, and it has both angioregulatory and immunostimulatory activity. So blocking Galectin-1 in vivo in the tumor microenvironment, we found that it promotes a reduction of tumor growth, a reduction of angiogenesis, microvessel density, and hemoglobin context, Importantly, it induces a reduction of VEGF receptor 2 phosphorylation, and also in combination with anti-VEGF eliminated resistance treatment, as we found in our previous study. Not only has this antibodies has an anti-angiogenic effect, but also an immunostimulatory effect, as you can see here in vivo after treatments with the anti-Galectin-1 antibody, a decrease of IL-10 in tumor-draining lymph nodes, and an increased frequency of CD8 positive gamma positive T cells. And this effect was recapitulated when we did the same experiment in MDAC5 knockout mice. So either when we block Galectin-1 expression or when we block glycosylated ligands, there is an increased stimulation of an anti-tumor T cell response. So when we were doing all these experiments, we were contacted by Jose Conejo Garcia. He made a very interesting observations that ovarian cancer patients, particularly 7.5% of ovarian cancers, are long-term survivors, and they share a single nucleotide polymorphism in TLR5 receptor. So one of the most striking observations was that in response in these particular patients, there is a decrease, significant and dramatic increase in Galectin-1 expression. So we started a collaboration with Jose, and what we show is that Galectin-1 links flagellated microbiota, IL-6 mediated inflammation, and tumor progression. So to make a long story short, in response to flagellated microbiota, TLR5 signaling induces an increase of inflammatory cytokines, particularly IL-6, which promote a recruitment of myeloid derived suppressor cells, and an increased frequency of gamma delta T cells that produce very high amounts of Galectin-1. So these are the cells that produce the highest amount of Galectin-1 within the immune cell compartment, and this contribute to abrogate anti-immune responses. So trying to search for potential receptors of Galectin-1, we found in collaboration with Marcelo Gil from Montevideo, from Uruguay, from Latin America, from Pasteur Institute. He's an expert in cation channels, and we found that TMEM176B, also called TORID, plays a key role in anti-tumor immune responses. So what we found was that by activating this particular transporter, there is a list of inflammasome activation related to a decreased influx of divalent cation ions, and this results in a reduction of the frequency of CD8 positive T cells and a response to immune checkpoint blockers. So when we block this particular transporter with BIK8644, there is an increase of NLP3 inflammasome activation, CD8 positive T cells, and an increased response to immune checkpoint blockers. So in summary, Galectin-1 functions as a glycopoint that links tumor immunity and angiogenesis. It promotes tumor immune escape. It induces 1, T18, and CD8 positive T cells via glycosylation-dependent mechanisms. It delivers tolerogenic signals to dendritic cells and myocytes expansion of regulatory T cells. It promotes polarization of macrophages to an M2 profile. I didn't speak about this particular work. It links tumor hypoxia to angiogenesis and activates VEGF receptor signaling, receptor 2 signaling, preserving angiogenesis in anti-VEGF resistant tumors. Moreover, it can link commensal microbiota to tumor-promoting inflammation. But Galectin-1 is not the only galectin, and other laboratories showed that other individual members of the galectin family can bind to distinct immune checkpoint receptors. For example, Galectin-9, this is a work from Vijay Kuchru. He showed that Galectin-9 can interact with TIM3, and Elizabeth Jaffe found that Galectin-3 can interact with LAG3. So it seems that different galectins act as soluble ligands of different immune checkpoint receptors. And they behave as glycosylated receptors, as glyco-checkpoints by themselves. So not only galectin-1 promotes angiogenesis, also other members of the galectin family. And this year we found that adipose tissue can promote angiogenesis by expressing high amounts of Gal-12. And in this particular study, we found that Gal-12 interacts with fucosylated antigens. So this is an exception. So different members of the galectin family contribute to angiogenesis, and we need to take this into account. Also, different members of the galectin family also contributes to immune escape. So we focus in this talk, we focus on 3 different hallmarks of cancer as those proposed by Hanahan and Weinberg: avoiding immune destruction, tumor promoting inflammation, and inducing angiogenesis. But galectins, and in particular galectin-1, play key roles in all the 10 hallmarks of cancer. And this is what we try to integrate together with Romina Chidotti and Mariano Salatino in a study that we published this year that is called Sweetening the Hallmarks of Cancer. So as I mentioned, galectins are not the only glycan-binding protein that can be considered glyco-checkpoint. Also C-glex and also C-type lectins. So in collaboration with Juan Garcia Vallejo from the EU University, an expert in C-type lectins in Amsterdam, we found that glioblastoma cells express very high amounts of the TN antigen, which has GalNAc, and can be recognized by the C-type lectin macrophage galactose lectin MGL, promoting macrophage M2 polarization and immunosuppression in glioblastomas. So, but not all the glyco-checkpoints need a glycan-binding protein. An excellent example of a glyco-checkpoint that is critical for inducing the function of these cells is those published by Juan Cubillo Ruiz. You have a great talk of Juan Cubillo Ruiz previously, and we collaborated with these particular papers where they showed factors in ovarian carcinoma induce a reduced expression of GLUT1 transporters, reduced internalization of glucose, which causes a defective glycosylation. So this particular glyco-checkpoint is independent, apparently, of glycan-binding proteins. This defective glycosylation is activation of XBP1, splice XBP1, uses the expression of glucose transporters and glutamine influx. So low glucose and low decreased respiration, decreased interferon gamma production, and dysfunction of these particular T cells. So, as I mentioned, immune checkpoints are characterized by at least 4 different hallmarks. And the second hallmark is that not only that they are co-opted by tumors, but they also need to promote resolution of autoimmune and chronic inflammation. So, in different studies, administration of recombinant galectin-1 or genetic delivery express autoimmune inflammation in different models of arthritis, multiple sclerosis, uveitis, colitis, hepatitis, diabetes, and orchitis. This is work from our lab, part from our lab, and part from different labs. Also, endogenous galectin-1 the autoimmune, the exacerbated inflammatory response, as shown by galectin-1 knockout mice, which show enhanced susceptibility, arthritis, and as you can see here, also of PMBS-induced colitis. So endogenous galectin-1 plays a key role suppressing immune responses, pathogenic immune responses, mainly by killing selectively, as I mentioned, Th1, Th17 cells, but also promoting tolerogenic dendritic. Here we published a study in which we demonstrated that spontaneously galectin-1 deficiency leads to
Gabi.
Yeah.
We are losing you sometimes. Do you mind maybe turning off your video?
And also a Sjögren-like disease.
Gabi, maybe you can.
This effect was also observed in MGAT5 knockout mice.
Gabriel, it cuts, so maybe you go a couple of times ago and it starts again.
What about the central nervous system? In the central nervous system, and to make a long story short, cytokines such as TGF-beta and IL-4, astrocytes are the cells that produce the highest amounts of galectin-1. Galectin binds to core 2 glycans on CD45 and promotes the activation of microglia, M2 polarization, and reduction of neurodegeneration. Interactions for treating autoimmune diseases, we found that treatment with recombinant galectin-1 suppress the disease, but there is a problem, and there are several obstacles. We need high concentrations of recombinant galectin-1 to exert robust anti-inflammatory activity. Galectin-1 is highly unstable in inflammatory microenvironments, and the question was why? We wanted to detect a particular therapy. So we explored whether it was related to the monomer-dimer equilibrium, to the affinity of glycan interactions, or to sensitivity to adverse inflammatory microenvironments induced by different biophysical and biochemical parameters. And based on different mutations, we generated what we call the super galectin-1 variants. With low concentrations of galectin-1s, we can achieve superior anti-inflammatory activities. So one of the last immune checkpoints, hallmarks of an immune checkpoint, is that they contribute also to immune tolerance during pregnancy. And what we found was that in response to progesterone in first-term pregnancy, galectin-1 is released and protects the fetus from maternal immune attack by creating immunosuppressive microenvironment, tolerogenic microenvironments. We performed this with Sandra Blois and with Petro Arc. And we found that galectin-1 knockout female mice had increased fetal loss, and also that galectin-1 rescue fetal loss induced by stress. Also, we found that in collaboration with Roxana Ramhorst, that women with spontaneous recurrent abortion had high amounts of blocking anti-galectin-1 antibodies in their serum. So the last hallmark of cancer that we will mention is impairing resolution of microbial infections and limit allergic reactions. And I don't want to stop before cover that was focused on galectin-1 and infection that was performed in collaboration with Elina Zúñiga and with her mentor Adriana Group. That Trypanosoma cruzi induces blocks pathogenic T cells by secreting galectin-1, and this is also the case of the bacteria Yersinia. Found that galectin-1 limits eosinophilia and allergic airway inflammation. So in this particular paper, I only want to mention that galectin-1 promoted chlamydia trachomatis infection, but dimeric galectin-1, but not monomeric galectin-1, highlighting the need of the cross-linking activity of dimeric galectin-1. So we observed this effect also in vitro, in vivo, in galectin-1 knockout mice, and also in MGAT5 knockout mice. So the last question was, all these effects that I mentioned are exerted through extracellular binding of galectin-1 to glycosylated ligands. So how galectin-1 is secreted? This was a mystery for many, many decades. It's secreted through a non-classical ER-Golgi independent pathway. So in collaboration, we collaborated with Vijay Ratimal from Yukon Health, and in response to LPS sensing, to intracellular LPS sensing, we found that galectin-1 was released as a danger-associated molecular partner or alarmin to control sepsis through mechanisms that involve non-canonical inflammasome activation, involving caspase 4, 5 in humans, and 11 in mice, cleavage of gasdermin D, and generation of pores by gasdermin D, promoting secretion of galectin-1. So non-canonical inflammasome activation, pyroptosis and necroptosis as well can be involved in the secretion of galectin-1. Also, we know that secretion of galectin-1 involves also the carbohydrate binding activity, carbohydrate recognition, and also released by exosomes. So this particular study related to non-canonical inflammasome activation will be published probably soon, and it's provisionally accepted. So I hope I convince you today that galectin-1 is a glyco-checkpoint that promotes tolerogenic dendritic cells, promotes dysfunction of Th1, Th17, and CD8 positive cells, regulatory T cells, polarizes macrophages and microglia to an M2 phenotype, links tumor hypoxia to angiogenesis, and links promoting inflammation and immunosuppression. I like to call that one the sweet case of Dr. Jekyll and Mr. Hyde. So when it's Dr. Jekyll, it suppresses autoimmune diseases, it inhibits inflammation, induces neurodegeneration, and anti-tumor immunity, promotes metastasis, and favors parasite infection. So based on this fundamental knowledge that we tried to build during these years, we generated to establish two platforms, two transfer technology platforms. One is a cancer immunotherapy and anti-angiogenic anti-galectin-1 antibody, and the second one is inflammation and autoimmunity, mainly based on the supercarbon protein. We generated doing preclinical studies to study safety and toxicity with Hugo Ortega in Isibet, Esperanza, Santa Fe before starting phase 1 trials, and we're also doing clinical validation of all these findings. Finally, I want to thank all the members, the current members of my group for all the hard work, the former members of my group, my wonderful collaborators, my wonderful mentors. I want to continue remembering always my mentors, Clelia Riera and Carlos Landa, and the agencies, the national agencies and the international agencies, and also the families that supported our work. So thank you very much, and I am open for all the questions you want. Thank you very much, Elena. Thank you very much, Gabi.
Thank you, Gabi. Thank you so much for sharing your journey of discovery of the galectins function and your efforts in translating them. We very much appreciate that. Now Elena will remind us how to ask questions, but I'm sure she has some comments first.
Yes, no, just want to thank Gabi. That was great. It was a pleasure to listen to all your discoveries, Gabi. Thank you so much. Really impressive. Okay, we will have Q&As via Twitter, and so if you'd like to ask questions for Gabi, please search for the account Global Immunotalks, find a tweet that says ask questions for Dr. Gabriel Rabinovich here, reply to that tweet with your question and mentioning the hashtag Global Immuno, and Gabi will use his own personal account, Gabi Rabi, to answer the questions. And yeah, thank you again, Gabi, and thank you for the extra effort to answer the questions via Twitter. It was a pleasure to have you and listen to you today. And thank you everyone for joining, and see you next week.
Yeah, thank you, Gabi, and thanks everybody.
Thank you very much. Thank you very much, Elena. Thank you very much, Carlos. Thanks so much.